morning. And first of all, I want to thank the organizer for this great meeting and for the kind and generous hospitality here. It's a great time. So yeah, now we'll shift to something completely different. Still microbiome, but this time we're shifting into the ocean microbiome. And the main passion of our lab is to understand how microbial interaction, especially host pathogen interaction in the ocean, can really shape the ecology and metabolic landscape at sea. And the main challenge is, for us at least, to try and bridge the scale. We'll see some example of how we can really zoom in into the single cell level, but also we want to shift to population and eventually also to go out to the field and understand this interaction in the real world. So the main phenomena that we study for years is algal blooms in the ocean. This is outstanding phenomena. This is the basis of life in the ocean. Similar to the terrestrial system, these algal blooms are responsible for 50% of the global photosynthesis on Earth. So hugely important. We see how much carbon they can fix in photosynthesis. But indifferent from the terrestrial system, of course, we're talking about single cells. We study these single cell bloom forming algae, eukaryotic algae, that form these massive blooms. Indifferent again from the terrestrial system, that we're talking about turnover time of tens of years, of course. In the marine environment, we're talking about turnover times of weeks of these massive blooms. And that's why we aim to study the molecular mechanisms that control cell fate in this bloom. When these massive blooms that stretch over thousands of square kilometers will crash or decline, this is a major metabolic valve to fuel the entire ocean microbiome. So we are, in the next half an hour or so, we'll zoom in into this process and see how different mortality agents, different pathogens regulate this key point in a marine food web. The way we do it, in a very simplified manner, in this slide here, we're moving between the balance between the complexity of the ecology by going out to scientific cruises. We bring back isolates of this algae and its pathogen. And there are many ways to die in the ocean in algal blooms. And today we'll hear mostly about algae virus interaction that regulate, but also we're expanding into understanding pathogenic bacteria, grazers, that's the one that eat them, and with special emphasis on the molecular basis for it, the molecular mechanism that mediate pathogenicity, and the metabolic consequence of such. Eventually, if we know specifically how of each of these pathogens signal and regulate the fate of the cell and what the metabolites that are being produced and how specific they are, because every pathogen will remodel the host in different manner, we can then use, similar to the pathogens in human and so on, we can use it as a sensitive metabolic biomarker to go back to the field and really look at interaction on a more functional manner. So let's talk about marine viruses, which is a very hot topic in marine microbiology for the last 20 years or so. This is how it will look like if you take a drop of water from basically anywhere in the aquatic ecosystem, and you simply stain it with a fluorescent dye for DNA, cyber green here. The big ones are marine bacteria, but the little ones are marine viruses, estimated to be around 10 million viral particles in a drop of water. So marine viruses are not only the most abundant biological entity at sea, but also major drivers of the nutrient recycling, and of course a huge evolutionary driver, shuffling genes between organisms and so on. But this is a very static view on viruses. And thus, we are very interesting to move to more functional manner and to look at the infection from within the infected cell in order to understand infection dynamic. One of the key ecosystem pathway proposed more than 20 years ago is the following. It's called the viral shunt, we talked about the massive amount of carbon fixed by these algal blooms, and where they are infected by viruses during lytic phase, this viral shunt can shift completely, in a very simplified manner, can shift completely the fate of carbon and other nutrients away from higher trophic levels. And as I mentioned, can fuel the ocean microbiome. So in the lab, we try to kind of demystify this proposed ecosystem pathway and really add more quantified manner, and also to understand the unique metabolic landscape that is produced by a viral infection that causing bloom demise. The model system that we're using extensively is between this single cell here. They have the fancy name Emiliania Huxley. They are major bloom forming. You can see here they form massive blooms in the North Atlantic, stretching over thousands of square kilometers. This is off the coast here of Norway. And they're not only hugely important as photosynthetic microbes, but also they are decorated by this beautiful calcium carbonate shell. So very important to the carbon cycle. And they are infected by these giant viruses. These viruses contain almost 1,000 genes, and that's another major discovery in aquatic virology in the last decade. Many giant viruses containing genes never seen before in any other viruses. So really blurring the boundary between a host and a virus. So we can put aside the textbook of phage biology, and that's very interesting now to learn more about the life cycle of these giant viruses. In our case, this virus is highly specific to this bloom-forming algae, and it contains not only close to 800 genes, but very unique pathway to biosynthesis, almost the complete biosynthesis pathway for sphingolipids that every eukaryotic cell will have, but this is the only virus known to date to contain such a pathway. Another major ecological and biogeochemical consequence of the infection is not only on the carbon cycle. You see here the famous Cliff of Dover in England. That's a kind of geological relic of this bloom demise over geological timescale because of this exoskeleton calcium carbonate shell. They're also hugely important for the sulfur cycle. With this single cell dye, they release gas, very famous gas, because it's the smell of the sea that we all love. It's called dimethyl sulfide, organosulfur. And it can go up, you see the numbers here in teragram a year. It can go up to the atmosphere from these massive blooms and really change cloud condensation nuclei. So amazing how the fate of a single cell can really change not only the carbon cycle, but also ocean atmosphere feedback. A few years ago, we actually found the enzyme in this algae that is responsible for this DMS production. And this DMS molecule is also the most ubiquitous chemical signals in the sea, mediating trophic level interaction all the way to predator-prey interaction. So very interesting, again, be able to link between the microscale and the large scale in the ocean. In the last 5 years or so in the lab, a major emphasis is to be able to disentangle or untangling the virus cell, the infected cell, to its different phenotypic component. As I mentioned, the virus cell, the infected cell, will have a major remodeling of the metabolism of the cell. So we're looking at it as kind of different entity. But then many other phenotypes can be produced. Most of the cell will die in a programmed cell death manner, but there's always less than a percentage that will survive the infection. So we study also mechanism of resistance. But if we keep the usual RNA-seq bulk information, we're averaging the entire phenotype here, and we want to try to disentangle them. So we applied several single-cell transcriptomic approach to be able to describe the different phenotype across the infection dynamic. Some of the approaches we used is either microfluidic, automatic microfluidic. Here we see capturing a single cell algae and then flash it with the reagent, or we can use flow cytometry-based sorting, so we can link it to unique physiological parameters. And as we saw earlier in the conference, also we're using the 10x genomic platform. And also we're looking at specific genes using single molecule FISH in order to look at both host and viral gene in a single cell manner, and also to visualize it. That gives us the powerful tools to answer even the most simplest question. In a given population in the ocean, what is the fraction of infected cells at a given time? Which is very, very difficult to quantify, and also in order in the future also to implement into ocean ecosystem models. But also we want to study these giant viruses in the natural environment, which is extremely difficult. And also for us is a new way to discover pairs of host and virus, because we're looking at active infection, RNA of both the host and the virus on a single cell manner, meaning they meet each other, infect, and replicate. So it's a way to capture interaction in action of every host pathogen interaction. Just as an example how it will look like, this is data from single cell transcriptomic. We're looking only on the viral genes. Each dot here is a single cell and the expression of the viral transcriptome. And we see this very nice heterogeneity. This is just a simple culture infected after a few hours harvested. So we're using this heterogeneity in order to understand why these cells are highly infected, where these cells are completely non-infected in isogenic culture in the lab, in order to understand mechanism of resistance, and from the other end, how the virus take over the host. If we did bulk RNA-seq, we would get this 1 point averaging the entire phenotype without the ability to match between infection state and host response. And what we did here with this same exact cell bioinformatically, we reorder them, and we're making this meta-analysis. We're making these mini neighborhoods. It's called meta cells, and we can cluster them now based on their transcriptional program of the virus, which gives us now a completely different view than rather looking at either infected or non-infected. Instead, we see the continuum of infection, again, between the non-infected to the highly infected, which is more diverse. It helps us now also, and now we can define what is the infection state, which is here. So again, we reorder the single cell. Each column here is a single cell. Each row is a viral gene. And when we reorder them now based on the infection state, suddenly we unravel unique transcriptional program induced by the virus, as you can see here. And what we found is that we can, by this approach, using this single cell dual RNA-seq, we can really define the kinetic class. And we found that this cluster, for example, share this transcriptional binding site as the immediate early, and late genes. So we can really understand more about the essential cascade of transcriptional program in the infection. It's also powerful, as I mentioned, because then we can also match directly between unique infection state on the single cell level to what the host does, right? And what we found, again, ordering the cell by the viral genes, this is now the host genes of the same single cell, and we found this very rapid shut-off of the host transcriptome, very similar to what was recently found in COVID. But we also found that it's highly selective. There's a hierarchy in the shut-off of the host, When it's nuclear encoded, we see this major shut-off, But when we look at the mitochondria encoded, we see stability during infection and also in the chloroplast until this point that it drops. So there's hierarchy in the shutoff, and we think in part that this is how these giant viruses fuel bioenergetically the infection by using the chloroplast and the mitochondria transcriptome. The other approach on the single cell level, as I mentioned, is single molecule FISH, where we can probe specific genes. This time we use the host photosynthetic genes as a metabolic marker, and from the viral side, more the capsid genes, and we can now follow the infection, both in microscopy and visualize where it happened in the cell, but also we can do it in high throughput in flow cytometry and image stream. It gives us now also a powerful tool to go to natural population and to zoom in into specific host and virus pair in a mixed population, which is still highly challenging. This is how it will look like in the microscope between early and late in the infection, using the probe for the virus RNA and for the host metabolic gene. We see here this shining population from viral expression after 24 hours during the lytic phase of infection of this algae. We can, as I mentioned, also do flow cytometry. And what's nice about it is that we can now follow the infection, and in this case, the fraction of infected cells and how it evolved during the dynamic, and also looking at the heterogeneity between the cell in a very nice way. This is from the viral gene expression, and this is the host one. So we can start to describe the dynamic and the different cell fate within within the population on a single cell level. Now, the big challenge is to using this single cell tool and move to the marine environment. We induce these massive blooms here in the fjords of Bergen in Norway in a setup that in ecology it's called mesocosm. Basically, we are not interrupting with the natural population. We inoculate this 11,000 liter bags. We seed them with the inoculum from the fjord, and we simply adding nitrogen and phosphate and let it evolve over a month. What you can see here very nicely, you see the milky color coming from the calcite shell of this single cell. We got massive, and here in the electron microscopy, we got massive blooms being induced, and now we can compare. What was very interesting, and again, we're not adding the virus, we're using, everything is the natural microbial interaction that coming from the fjord water as a seed. What was very powerful here is that some bags crash because of viral infection, and this is viral production, but some crash from other reasons. So we have a nice comparative way now to really understand the infection dynamic and its impact on the metabolism. Very interestingly, and that's good work in the lab now, every dot that you see here is a single cell transcriptomic, and we're looking at the viral genes, again, on the transcriptome level, so we can start really in an ultra-sensitive manner to look at the infection dynamic on a single cell manner and detect the infection at very, typically, we will look at this phase, right, on the demise phase, but we can start to look at the infection very, very early on in the dynamic of the bloom formation. So watching the pandemic in action. It can give us now interesting approaches also to look at the bystander cell and to see if there's interesting chemical communication between them. And here again, we applied the single molecule FISH to look at viral infection and the fraction. And here came a very big surprise now that shifting a lot of what we do in the lab, see what happened. If you look extracellular, as was done in aquatic virology for the last 50 years, this is the bloom crash, the algal crash. The bloom crash very nicely and sharply in a synchronized manner. And this is where you count these giant viruses extracellularly. They are so big, these viruses. They are so big that we can count them in the flow cytometry like bacteria. So this is the dynamic of the viral infection from outside the cell. But let's look inside the cell, and that came the big surprise. Although we are in the demise phase of the bloom, only 30% of the population were actually infected by these viruses. So how come they all crash in a synchronized manner? There are different possibilities that the infected cells, for example, release chemicals that we study them now using metabolomics to understand how they affect the neighboring cells, other predators or parasites, and also maybe pathogenic bacteria, the microbiome associated with the bloom. We also characterize it very extensively. What Noah in the lab found independently were isolating from bloom demise in the Atlantic Ocean that actually these blooms contain, towards the end of the bloom, pathogenic bacteria from the Rosobacter clade. And what she showed is that they have two phases, a coexisting and a pathogenic one that can cause the algae to die. But this is the bacteria, and we're talking about the viruses. So what's going on here? Before that, this is a new paper by Noah that she showed that the pathogenicity and the shift between coexistent to pathogenicity or virulence is very strongly dependent on metabolites that are released by the host on different physiological states. We found that different metabolites, in this case benzoate, was essential for coexistent, and DMSP, that's the precursor for the same DMS that we talked about in the beginning. This algal cell can accumulate this precursor up to hundreds of millimolar in the cell, but when they start to get to stationary phase, they become leaky. So they release this molecule that can induce virulence of the bacteria. But that's the bacteria and the alga, and before we talked about viruses and the algae, are they connected somehow? So we started collecting microbiomes from different blooms, and we preserved them, and then we revived them in the lab, and start to look at tripartite interaction, where we have isolates of the algae, its specific giant virus, and also microbiome that associate from the blooms, and also the molecule that mediate them. What we found, and that's the new stuff now in the lab, which is very, very intriguing for us, is that we have the three together. We get synergy in killing, as you see here. This is a typical cell death and crash of the algal when it's just the virus, but here it's when the two exist together, and now we have also isolates that are pathogenic, and the new thing in the lab is to try to understand how viral infection and the chemical molecules released by viral infection and metabolites can induce and prime virulence of the bacteria. So acting in synergy in a tripartite manner. Lastly, in the last part, the last 3 minutes of the talk, is we're looking at the unique metabolites that are released by viral infection using different metabolomics approach. As I mentioned, the virus encode for unique sphingolipid pathway. So we're using a lot of comparative lipidomics in order to understand the unique lipids that are, as you see here, strongly induced during infection. But also we're using imaging mass spectrometry. And in this study here, this is plaques. We know how to do plaques from the 40s of previous century. But here we mixed it with a MALDI mass spectrometry imaging. And we're looking at the same plaque here by, if you have a glass for each of the metabolites, each nice figure here is a different metabolic landscape of each of these metabolites. And that enables us to identify unique metabolic bioactive molecules that are unique to viral infection, because what's the beauty of the plaque, starting from a single cell in the center of the plaque and then propagating, is that in one snapshot, you get infection state in a gradient from the center to the periphery where it stops. So we start to generate this metabolic landscape images. And of course, it will have major impact when this cell lies on the metabolic footprint in the water. And for this, I have 2 minutes left. Sorry. For this, we did extensively during the bloom you saw before, exometabolomics approach to profile the unique metabolites here at each phase of the bloom, to start to describe the dynamic, the biology of the bloom by the molecules that are released into the water. And what we found is that, and from all these 7,000 mass features, we wanted to distill the unique molecules that are unique to viral infection. So we correlated to unique viral gene and metabolic biomarker, and what we found is these unique 20 molecules, which are all highly halogenated molecules. They are chloroiodo metabolites. This is heaven for the natural, we realized, the natural product community, because many of these molecules are important bioactive molecules, antiviral and antibacterial. When we shifted now to the lab in order to isolate this molecule for the activity, because in the marine environment, the signature was so strong in the water, but we want to understand why. So we're shifting to the lab, and for a year, we couldn't detect this molecule because we used just the algae and the virus. Until this day, until this day came where we added the microbiome of the bloom. And again, the tripartite interaction are essential to produce this unique chloroiodo organometabolites that now we aim to understand in this tripartite interaction, its bioactivity and mechanism of biogenesis. So that's my last slide. So we started with understanding on and quantify the viral shunt, but now we're shifting into the pathogen shunt, where two pathogens can act in synergy, and the unique metabolites that are released by viral infection can prime other members of the microbiome community, induce their virulence, and they can act in synergy. So now we're very much interested to understand how it will change the carbon and sulfur, what will be the ecological and biogeochemical consequence of such a synergy that was never described before in the marine environment. A good place to stop and to thank a great lab member. I'm so fortunate to have them working with me for the last years. The funding to all of you, and this is how we normally look like. So thank you very much.